Hello, I'm M.Collin from Newcastle University, and I'm going to give you a brief summary of some new research published in Science Immunology about the origin of LCH lesions. So this is a study by Egla Kvederaiti, Florent Ginu, in collaboration with myself and Paul Milne in the lab, and many other authors who've contributed. Now, the main question of the research is how do you make an LCH lesion? And there are several ways to approach this question, but it all starts with, we think, cells in the blood that have mutations. So you're probably aware that there are mutations in certain genes, such as BRAF, that cause Langerhans cell histiocytosis. And one of the things that have been done is to look at the different types of white blood cells that might contribute to LCH lesions and to test them all for the presence of the mutation. And what we and most other groups find when we look at dendritic cells and related cells called monocytes, that there are mutations in certain fractions of these cells. So the system's a little bit complicated, and we don't need to worry too much about these names here, but there are various different flavors of dendritic cells illustrated by these different colors. And what we find is that most of the mutation indicated by the red star is found in these cells called DC2 and DC3, and also in monocytes. Monocytes are the cells that turn into macrophages inside tissues. So there might be a little bit in DC1, but these cells are very rare, and it's quite difficult to examine that. And sometimes we find a little bit of mutation in these PDCs. P stands for plasmacytoid. So we think that most of the mutation is present in these red and orange and black cells, the DC2, DC3, and the monocytes. So that tells us potentially which cells could make an LCH lesion, because it's the cells with the mutation that go on to form the lesion. But then to actually tie that all together, Egla and Florent started looking in the other direction using some very high-tech methods, and that was to take all the cells of the lesion apart and then ask the question, can you tell from the characteristics of the lesional cells where they might have come from? And I would also suggest you look at this press release from the Karolinski, which describes the paper. This is Egla here, working in the Karolinska, and also Florent, one of the PIs who divides his time between Paris and Singapore. So what they did was to take microscopy, some very high-tech microscopy where you can look at lots of different markers on the surface of the cells, and then you can also divide up the images and do some quantitative, so you can measure the different proportions of markers on the surface of the cells and determine their relationship to other cells. And then he also took the lesions apart into single cells and looked at the genes that were expressed inside each of those single cells. And essentially, these two approaches indicate that there are two dominant signatures within the LCH lesion, one that corresponds to this DC2 cell, and another signature that corresponds to either a DC3 or a monocyte. These cells are quite closely related, so it was difficult to separate those two, but there were basically two signatures, a DC2-like and a DC3 or monocyte. And you can see on the left here this very complicated looking lesion with all these different types of cells, but when you classify them, you essentially have two types of LCH cells, the dark purple ones and the blue ones, which are indicating the two different origins from DC2 or DC3 monocyte. And this plot that looks like a bit of a map on the right-hand side here, again, shows every dot is a single cell, and they either have a relationship with DC2 in the pink or DC3 monocyte in the blue. So what this suggests is that all of these cells, DC2, DC3, and monocyte, work together to make the LCH lesion, but each type of cell contributes to a slightly different flavor of LCH cell once it's in the lesion. And we can see that from evidence of the markers that they express within the lesion and the expression of the genes within each cell. So how does this happen? And in order to understand this a bit better, we went back to some work published by Caroline Hutter at the St. Anna Hospital in Vienna. And she previously showed that there was a system known as Notch, which is a signaling system between cells that was active in LCH lesions. So this red staining in the middle panel shows the expression of CD1 showing Langerhans cells, and you can see the brown in the middle shows the expression of Notch, indicating that there is Notch signaling active in LCH lesions. So we wondered if Notch might play a role in this conversion of the blood cells into the Langerhans cell lesion. And what Paul in the lab was able to show was that there was a communication between the two dominant groups of cells within the lesion. So the DC2s, which readily express Langerin when you culture them in vitro, they also express Notch ligands, so things that can give a Notch signal to these other cells, DC3 and monocyte. And these cells are a little bit more fussy in their requirements for the expression of Langerin and forming Langerhans cell-like cells, and they require a Notch signal. And what we found is that the DC2s can provide this Notch signal to the DC3s and the monocytes. And this just shows some of Paul's microscopy showing the famous Birbeck granules in these different types of cells. So they all potentially look like LCH cells after they've had this Notch signal, but they can come from each of these different sources, the DC2, the DC3, and the CD14 monocyte. So it looks like DC2 is the kind of instigator, and it brings in these other cells, DC3 and monocyte, and induces them also to express the LCH phenotype. So what does this mean for patients? Well, it basically means that this signal, this Notch signal, might be a critical part in the formation of the lesion, extending the observation that Caroline made a few years ago. And potentially we might be able to interrupt this signaling pathway. And if this was a critical thing in the formation of the lesion, then interrupting this signal would lead to the resolution of the lesion or prevention of new lesions forming. And that's why we think it's potentially useful to patients. Okay, thank you.